The title of the article is Scientists Reveal How Signals from Pathogenic Bacteria Reach the Inflammasomes Within the Cell. So this means there's a vital protein caused by a signaling protein interferon, which is required to activate the danger-sensing proteins within the cytoplasm of a cell. So these crucial proteins are known as inflammasomes, like explained. These are your danger sensors for things such as infection and disease throughout the cell. So without inflammasomes, our body has no rapid immune system, so there's no way to defend ourselves. So by knowing this key information, it opens up a new gateway for the scientific community and medicine to make more antibodies to defend against and protect the body against such pathogens. That's it.
Receptors are an organ or cell that are able to respond to light, heat, and external stimuli and transmit a signal to sensory nerves. An example of a receptor can be shown in this lock and key model.
Alright, so the most basic method, or one form of the receptor model, would be the lock and key model or method. And you would have a substrate and an enzyme which lock together with interlocking mechanism right here at the active site. And together these would form a complex. So if you put the two together, you're going to have a substrate enzyme complex. And that's your most generic form. Okay. So over here we have an example of how the immune system will be compromised in the human body. So as you can see, these are the receptors here. They have a very specific shape which need to latch on with the stimulus. So the way a pathogen or bacteria or virus can get into your body will be taking shape of the same receptor and then going into that receptor and inhibiting it and getting into your body, compromising your immune system.
Chemoreceptors are the type of receptors that detect the levels of CO2 in the blood by monitoring the concentrations of hydrogen ions in the blood. An example of chemoreceptors at work include a person is hyperventilating from an anxiety attack. This causes an increase in ventilation, which causes too much CO2 to be removed from the body. Without CO2, there is less carbonic acid, and the blood causes the hydrogen ions to decrease and the pH to rise. In response to these changes, chemoreceptors detect the change and send a signal to the medulla, which signals the receptor muscles to decrease ventilation so CO2 and pH can return to normal levels.
Bringing it back to the article is toll-like receptors. A small number of toll-like receptors can detect a broad range of human pathogens, as well as a variety of other molecules that indicate tissue damage by a process called pattern recognition. These receptors indicate two arms of the immune response, the innate and adaptive responses that work together to fight infection in mammals. The innate response provides immediate protection. However, it is relatively nonspecific in its mode of attack on pathogens, which results in a damage to healthy tissue if the innate immune response lasts too long. The adaptive response, on the other hand, generates antibodies secreting B cells and cytotoxic T cells that are specific and efficient at targeting pathogen. Unfortunately, this process takes too long to develop than the innate response. Because toll-like receptors function as first responders to danger signals, they are certainly significant in research efforts to combat infections and inflammatory diseases. New strategies for manipulating immune response depend on understanding the cell biology of toll-like receptors, including their structure, cell localization, signal transduction pathways, and expression patterns.